I'm Melody, and the project I'm presenting at the CURF 2022 Fall Research Expo is titled Impaired Fast Spiking Interneuron Physiology in a Mouse Model of KCNC1 Epilepsy. So let's get started with some background info. The KCNC1 gene encodes for a subunit of voltage-gated potassium channels called Kv3.1, and KCNC1 mutations can be gain or loss of function, either of which can alter the unique kinetics of Kv3.1 channels that allow neurons to fire at high frequencies. Now, voltage-gated potassium channels play a key role in neurons' ability to repolarize and terminate action potentials, and Kv3.1 channels are particularly prevalent in inhibitory interneurons. And if they can't properly regulate the excitability of surrounding cells, this could lead to neuronal hyperexcitability and seizure activity. And I investigated this potential mechanism of KCNC1 epilepsy using an experimental mouse model with the A421B variant involving a loss of function mutation. And this leads me to my research question. How does the KCNC1 A421B mutation affect the intrinsic physiology of PV interneurons? And on a larger scale, how can Kv3.1 dysfunction lead to neural circuit hyperexcitability and developmental and epileptic encephalopathy? So, to measure the intrinsic physiology of PV interneurons, which are a specific type of fast-spiking inhibitory interneuron, I surgically removed brains from wild-type and KCNC1 mice and incubated brain slices at physiological temperature for half an hour. From there, I used a technique in electrophysiology called whole cell patch clamping, which allows me to directly measure the activity of PV interneurons located in layers 2 to 4 of the cortex. Now, after analyzing this data, I found that there is a clear difference in the intrinsic physiology of PV interneurons in wild-type and KCNC1 mice. So, compared to wild-type PV interneurons, KCNC1 PV interneurons have a significantly slower downstroke velocity, which may help to account for their significantly higher AP amplitude in APD50, as well as a tendency to undergo depolarization block, which helps to explain their significantly lower AP frequency. In other words, the rate at which KCNC1 PV interneurons repolarize is much slower, and as the amount of current injected into the cell increases, KCNC1 PV interneurons can enter a state where no more action potentials can be fired, no matter how much the cells are stimulated. And it's these factors that may contribute to larger, longer-lasting action potentials. Now, putting this into perspective, it's possible that the KCNC1 mutation can alter the unique biophysical properties of Kv3.1 channels, and this leads to a major reduction in potassium current density that impairs the ability of fast-spiking interneurons to inhibit excitatory cells, resulting in levels of neuronal hyperexcitability that could cause seizures. With that in mind, here's everyone I'd like to thank for making this project possible, and thank you for watching.